Hi viewers, today we are going to discuss the monoclonal antibodies. Under this topic, we are going to discuss introduction to the topic, history of the monoclonal antibodies, principle involving in the hybridoma technology, procedure, protocol, uses of monoclonal antibodies, clinical use of monoclonal antibodies and the applications of monoclonal antibodies. Let us start with the introductory part. Antibodies are protein molecules or immunoglobulins produced by a specialized group of cells called B lymphocytes, otherwise plasma cells in all the vertebrates. Antibodies are a part of defense system to protect the body against the invading foreign substances, namely antigens. Each antigen has specific antigenic determinant site on its surface, which is known as the epitope. The antibodies have complementary determining regions, CDRs, which are mainly responsible for the antibody specificity. That is, the epitope of the antigen as the complementary determining region in the antibody, that is nothing but the paratope. In response to a multivalent antigen means an antigen with different epitopes. Let us assume epitope number 1, epitope number 2, epitope number 3 and epitope number 4, like this. This is the multivalent antigen. In response to a multivalent antigen with different epitopes, B lymphocytes produce many different antibodies. All these types of antibodies which can interact with the different epitopes on a multivalent antigen are said to be as polyclonal antibodies. All these types of antibodies they form the polyclonal antibodies. Polyclonal antibody response facilitates the localization, phagocytosis and complement mediated lysis of the antigen. Direct biochemical purification of monoclonal antibody from a polyclonal antibody mixture is not feasible. Antibodies produced by a specific clone of lymphocytes that act against a single or specific epitope of a multivalent antigen. It was a dream of scientists to produce monoclonal antibodies for different antigens. Animals were immunized against a specific antigen. B lymphocytes were isolated and cultured in vitro to produce monoclonal antibodies. This approach was not successful since culturing of normal B lymphocytes is difficult and synthesis of monoclonal antibodies was short-lived and very limited. Then coming to the difference in between polyclonal and the monoclonal antibodies. Polyclonal antibodies cheap to produce and whereas monoclonal expensive to produce. Polyclonal mixed population of antibodies, here monoclonal single antibody species. And polyclonal antibody may bind to different areas of the target molecule, whereas monoclonal antibodies will only bind single specific site. Polyclonal antibody tolerate of small changes in protein structure, whereas monoclonal antibody may recognize a particular protein form. So these are the various number of different kinds of antibodies on a single multivalent antigen. Here this antigen bound by a single type of antibody, monoclonal antibodies. And coming to the history of the monoclonal antibodies, in 1975, George Kohler and Caesar Milstein devised a method for preparing monoclonal antibodies, which quickly became one of immunology's key technologies. This is known as the Hybridoma technology, and for this they were awarded Nobel Prize in the year of 1984 for the physiology or medicine. And afterwards, in 1988, Greg Winter and his group pioneered the techniques to humanize the monoclonal antibodies. And coming to the principles of the hybridoma technology. What is hybridoma? Is nothing but a somatic cell hybridization, forming a hybrid cell by fusing two different somatic cells. For this, what we are doing, a normal activated antibody producing cell, a normal activated antibody producing B cell is fused with a myeloma cell. Myeloma cell, it is nothing but a cancerous plasma cell to produce a hybrid cell. A normal activated antibody producing B cell is fused with a myeloma cell. Myeloma cell is nothing but a cancerous plasma cell in order to produce a hybridoma cell. Oma is the suffix of cancerous cells. This hybridoma cell retains the properties of both the parent cells, that is, antibody secretion property of the B lymphocyte or B cell, and uncontrolled division property or immortal growth property of the cancerous cell. The clone of hybridoma cells always produce a particular type of antibodies that react with the specific antigens. Same kind of antibodies are produced from the clone of the hybridomas. So a hybrid, it is a cell produced by the fusion of two different kinds of cells. Oma, we will use the suffix for the cancer related terms like the lymphoma, adenoma, carcinoma, sarcoma. Like that, it is a hybridoma. The B lymphocyte with immortal growth property. Then coming to the procedure for the production of the monoclonal antibodies. What we will do, we will immunize the mice with a selected antigen. A mouse is injected with a foreign protein or antigen. And later on, after few days, remove the spleen from the mice. Remove the spleen from the mice and collect the antibody producing B cells. Collect the antibody producing B cells. Then followed by collect the tumor cells or cancerous cells, myeloma cells from the tumor of a mice, otherwise from a culture. And both the cells are get fused together. Both the cells, they get fused together. Thereby, some of the mouse cells fuse with the tumor cells to make hybrid cells called hybridomas. Here in the diagram, we are observing these are the hybridoma cells, and these are the mouse cell and tumor cells are fusing, and at the bottom we are seeing the unfused cells. Here, for the fusion of both these cells, that is B lymphocyte cells and as well as the cancerous plasma cells, for the fusion, both the B cells, myeloma cells are mixed in the presence of the fusogens. What do you call them? Fusogens. This is generally it is a chemical glue which is called as the polyethylene glycol, PEG, polyethylene glycol. And instead of PEG, previously they were used Sendai virus. Instead of polyethylene glycol, previously scientists used Sendai virus for the cell fusion. But nowadays, the chemical glue which effectively fuses both the cells is the polyethylene glycol. It is very, very important. Then the mixture is then washed and placed on the selective medium where only hybridoma cells can grow. The mixture of cells placed in a selective medium that allows only hybrid cells to grow. So this selective medium, the medium used for the identification and selection is a HAT medium, HAT. HAT stands for H for hypoxanthin, A for aminopterin and T for thymidine. Hypoxanthin, aminopterin and the thymidine mixture. It allows only the hybridoma cells to grow in that medium, and remaining unfused cells, they will be not grown. Here spleen cells and hybrid cells are HAT resistant, but myeloma cells die due to the HAT. Spleen cells can live for 1 or 2 days and then die. During cell fusion, it's very, very important. During cell fusion, for example, in the mice, there are 20 pairs of chromosomes, 20 pairs and the 20 pairs in both the B lymphocytes and as well as the cancer cells. Due to the fusion of these two cells, a hybridoma cell has been formed which contain 40 pairs of chromosomes instead of normal 20. What happens when the chromosomal number is enhanced? During the cell fusion, heterokaryon of B cell and myeloma cells formed, followed by random chromosomal loss. It is very, very important process where the heterokaryon containing both the nuclei of B cells and myeloma cells, although the chromosomal get doubled rather than the 20 pairs, then the heterokaryon formation followed by the random chromosomal loss. This leads to the expression of some normal B cell genes but grows indefinitely like a cancerous cell, and finally the chromosomal number brought to normal. Now hybridoma cells are selected and cultured for commercial production of the monoclonal antibodies. Clones are tested for the production of the desired antibodies. Whether the clones have been have the capacity to produce the desired antibody or not, we will check them. And the chosen hybridomas are injected into the peritoneal cavity of the mice. Otherwise, the hybridomas are cultured in laboratories and stored frozen until they are needed to generate additional supply of monoclonal antibodies. Separate pure monoclonal antibodies from the hybridomas by differential centrifugation. Here there is a diagrammatic representation. Here we are using an antigen with 4 kinds of epitopes. For each and every different epitope, the mice produce, whenever the mice immunized with these kind of multivalent antigen, then they can capable to produce different kinds of spleen cells. They are nothing but the B lymphocytes. Now these B lymphocytes are fused with the myeloma cells. For the fusion, we are using the polyethylene glycol, and thereby hybridomas have been formed. These hybridomas are cultured for the screening, and we are using the HAT medium, hypoxanthin, aminopterin and the thymidine medium, and thereby We are selected these cells as a successful hybridomas and they cultured in an appropriate medium to produce the clones of the hybridomas. These clones of the hybridomas they tend to produce different kinds of antibodies against to different specific epitope of a multivalent antigen. So here it is also the same thing, a multivalent antigen with the different epitopes injected into the mice, and here it is a polyclonal antibody serum, but we require the spleen cells and mixed with the myeloma cells. These are the hybridomas. They screened and cultured for the production of specific monoclonal antibodies. And coming to the protocol for the monoclonal antibody technique. Let us see the protocol for the monoclonal antibodies. First, we have to go for the immunization of the individual and isolate the cells and go for the cell fusion, that is fusion of both the normal B lymphocyte and the cancerous B lymphocyte. And here we will use the polyethylene glycol as the fusogen and go for the selection of hybridomas by using HAT medium, hypoxanthine hymenopterin thymidine medium. A screen for the successful hybridomas, select them and produce different clones, and we will go for the cloning and propagation of their population, and we have to check whether the monoclonal antibodies have been prepared against a particular desirable antigen or not. Characterization has been done and stored them as frozen antibodies. Whenever we require, we produce them in large scale. This is the protocol for the monoclonal antibody preparation. So these are the steps involved in that. Same thing. And uses of the monoclonal antibodies. Monoclonal antibodies are diagnostically useful in detecting chlamydia. It is a streptococcal bacteria. Then it is useful in detecting HIV, which causes AIDS. They are useful in measuring protein, toxin and drug residues in the body. Used for the blood and tissue typing. Used for measuring antibiotic residues in milk. Used in suppression of rejection reactions in organ transplantation technique. Neutralizing the exotoxins produced by bacteria. Inhibition of platelet clumping in the patients underwent angioplasty. Useful in treatment of autoimmune diseases. These are certain uses of the monoclonal antibodies. And come to the clinical use of monoclonal antibodies. Here we have taken into consideration some monoclonal antibodies and their curable diseases. Here trastuzumab used for the advanced breast cancer, used to treat the advanced breast cancer. Alemtuzumab, B-cell chronic lymphocytic leukemia. Gemtuzumab, acute myeloid leukemia. Rituximab, relapsed non-Hodgkin's lymphoma. Hybridumab, relapsed non-Hodgkin's lymphoma. So these are the certain monoclonal antibodies which are used extensively to treat these kinds of diseases. This is a list of monoclonal antibodies which are used as drugs to cure different kinds of disorders. So this is a list of some important monoclonal antibodies. Monoclonal antibodies used in COVID-19 treatment. See here, monoclonal antibodies are antibodies developed in a lab to help our bodies fight against the infection. There are of about 100 number of monoclonal antibodies approved by the FDA, Food and Drug Administration Authority, to treat health conditions such as cancer and autoimmune diseases. At least 5 monoclonal antibodies had been given EUA, emergency use authorization, for the treatment and the prevention of COVID-19. So this is the COVID virus, and these are the epitopes, spikes, and these are the monoclonal antibodies which are used to control the virus. See here, mAb and mAb. These are the monoclonal antibody injections which are used to treat the COVID-19. Along with that, tocilizumab and itolizumab. These are also used to treat the COVID-19 disease. And certain applications of hybridoma technology, serological applications, identification of ABO blood group, and diagnostical applications, detection of pregnancy by assaying hormones with monoclonal antibodies, separation of one substance from a mixture of very similar molecules, and coming to the immunopurification purpose, purification of individual interferons using monoclonal, inactivation of T lymphocytes responsible for rejection of organ transplants, and therapeutic applications, removal of tumor cell from bone marrow, treatment of acute renal failure, treatment of malignant leukemic cells, B-cell lymphomas, and variety of allograft rejections after the transplantation. These are the different applications in the fields of serology, diagnosis, immunopurification, and the therapy. So this is all about the monoclonal antibodies and their production, hybridoma technology, and their uses, applications of the monoclonal antibodies. Thank you one and all.